हेलो स्टूडेंट्स, आज अपन डिस्कस करेंगे बैक्टीरिया के रिप्रोडक्शन के बारे में। बैक्टीरिया जो होता है इट रिप्रोड्यूसेस मेनली बाय एसेक्सुअल मेथड एंड देयरफॉर दे हैव अ डोमिनेंट हैप्लॉइड फेस इन देयर लाइफ साइकिल। They do not have sex organs or gametes, but they definitely show genetic recombination, that is exchange of genetic material. नॉर्मली जो होता है ए सेक्शुअल प्रोडक्शन ही पाया जाता है इसमें। और ए सेक्शुअल प्रोडक्शन में सबसे पहले जो है वो होता है बाइनरी फिशन। ये जो डायग्राम आप देख रहे हैं बैक्टीरिया का, दिस इज शोइंग द बाइनरी फिशन। इट इज़ द सिंपलेस्ट एंड द मोस्ट कॉमन मेथड ऑफ मल्टीप्लिकेशन इन बैक्टीरिया। And under favorable conditions, the cell divide into two daughter cells by a transverse wall. During fission, the cell elongates and there is division of the nuclear material. In gram-negative bacteria, it is followed by the development of a simple median constriction that finally results in the complete separation of two daughter cells without cross wall formation. ये जिस तरह से आप फिगर में देख रहे हैं। In gram-negative bacteria, however, first a transverse cell membrane is laid down between the two nucleoids of a dividing cell, and then the centripetal growth of the cell wall forms a complete cross wall. The transverse wall between the daughter cells is very often incomplete, and then the daughter cells remain connected to the transverse membrane. When the planes of the successive divisions are parallel, long chains may be formed, and perpendicular to one another sheets or packets of the cells are formed. In binary fission, DNA replication precedes septum formation. The two resulting cells are mirror images of one another. Analysis of cell wall components of dividing cells indicate that the chemical constituents of the original mother cell wall are equally shared in the cell walls of two resulting daughter cells. Process of binary fission is very rapid. Bacterial cells may undergo fission every 20 to 30 minutes, and under favorable conditions, within 6 hours, approximately 250,000 cells may be formed from a single bacterium. But in nature, the essential nutrients are generally a limiting factor, and moreover, the accumulated waste products of bacteria also restrict their rapid growth. Normally, growth and multiplication is very fast in the early stages, reaching at the optimum and then declining rapidly. Now, if you compare the fission with the normal mitotic division, it is quite different, as the fission does not involve spindle formation during the division of nuclear material. It is not definitely known whether in fission the genetic material is equally distributed between the daughter cells. Here you can see the diagram. Now, next method of asexual production, it is the budding. In this type of multiplication, the bacterial cell gives out many outgrowths. These outgrowths are filled with cytoplasm, and other cell organelles also move into them. These outgrowths are called buds, and they eventually get detached from the parent cell by the constriction and each grow into a new bacterial cell. In this diagram, in the A diagram, you can see this is the budding bacterium. Buds are formed and cytoplasm get entry, and then it is get detached from the parent cell and the complete bacterial cell is formed. Now, budding differs from binary fission in many respects. During binary fission, symmetry of the cell with respect to the longitudinal and transverse axis is maintained throughout the entire process of division. In contrast to binary fission, in budding bacteria, most of the new cell wall components are used in the synthesis of the bud instead of being divided equally between the two progeny cells as in the binary fission. In budding, the mother cell retains its identity even after the formation of new progeny. On the contrary, in the binary fission, the mother cell loses its identity after the formation of daughter cells. Consequently, a primitive aging process occurs in the budding bacteria, which is absent in those dividing by binary fission. The other method is by the conidia. In this B diagram, you can see the conidia formation, just as in fungi you see. Many filamentous bacteria, example Streptomyces, form chains of small spherical spore-like conidia at the tips of the filaments. A conidium develops by the formation of a transverse wall at the tip of the filament. The filaments bearing conidia are known as conidiophores. After liberation, each conidium gives rise to a new filamentous bacterium, provided conditions for the germination are favorable. So, asexual reproduction normally takes place under favorable conditions. You can see these are the conidiophore or the sporophore, and then chains of conidia are formed. And again, each conidium can reproduce and form the entire filament. Now, other method is the cyst and the endospores, the formation of endospores. Endospores are intracellular resting cells and they differ from the cyst in the sense that in their formation only a part of original cell is utilized, while in the cyst they are found by the deposition of additional layers around the existing cell wall. That means the entire cell is transformed into the cyst, while an endospore, a small part of cytoplasm, is utilized and it forms the endospore. Gram positive among cocci and spirilla, endospores are formed only in few species. Typically, one endospore is formed in a vegetative cell. The cell producing endospore is termed as sporangium. The shape, size, and position of the endospore within the cell may vary in different species. Endospores are oval or spherical, and their diameter may be larger or smaller than that of the vegetative cell. Their position within the sporangium may be central, subterminal, or terminal. इस तरह से आप डायग्राम में देख रहे हैं कि ये सेंट्रल पोजीशन है, सब टर्मिनल है और टर्मिनल पोजीशन है एंडोस्पोर की. एंड मोस्ट ऑफ द एंडोस्पोर फॉर्मिंग बैक्टीरिया जो हैं दे आर सॉइल बॉर्न एंड फ्यू आर पैथोजेनिक ऑल्सो. And pathogenic spore forming bacteria causes disease by toxin production. How this endospore is formed? The ability to produce endospores is normally never expressed during the active phase of growth. It is expressed only when the vegetative growth reaches the stationary phase due to nutrient limitations. It is believed that sporulation is controlled by protein metabolism of the cell, and a spore is formed only when the vegetative protein of the cell is converted into spore proteins or when new proteins are formed. अगर हम स्ट्रक्चर की बात करें एंडोस्पोर की, this is the diagrammatic representation of the endospore formation. Here we will see this vegetative structure. And then the nuclear bodies, the initiation of transverse wall, complete transverse wall is formed. Then in diagram E, you will find formation of new cell wall around the endospore, and it get detaches from the cell wall of the mother cell F. And then G, you can see it is endospore, and in H that this endospore is released. Now this is the diagrammatic representation of endospore formation. If we come to the structure of endospore, the endospore consists of a central protoplast, the core, mainly composed of DNA, transverse RNAs, ribosomes, enzymes, and accessory factors. It is enclosed by a thin membrane, variously called core membrane, germ cell membrane, or inner membrane. The multi-layered region around the core is called as the cortex, and it is largely composed of calcium dipicolonic acid, peptidoglycan complex. Outer to the cortex, there is an electron-dense layer called spore coat, which is differentiated into an inner coat and an outer coat. The spore coat is mainly composed of proteins and accounts for 30 to 60% of the dry weight of the spore. Exosporium is there. Endospores are tough enough to last for years, enduring many stresses. Some of the spore producing bacteria are serious pathogens. For example, anthrax is caused by Bacillus anthracis, and its tough spores remain viable for years on the wool from the infected animal. The disease gets passed to weavers and other people through cuts in their hands. Now, if we talk about the germination of endospore, free endospores do not show any detectable metabolism. Endospores germinate under favorable conditions only, and sufficient nutrition, humidity, and suitable temperature are essential for their germination. These conditions trigger a series of metabolic reactions. Spores imbibe water and swell, losing their refractivity. The spore coat ruptures and new vegetative cells grow out. The spore wall forms the wall of the vegetative cell. Each bacterial cell forms only one endospore, which in turn gives rise to a single bacterium. Therefore, sporulation cannot be considered as a process of multiplication. It is in fact a method of perennation.